Hello, dear colleagues. My name is Yuriy Shevchenko, and I am the Vice President of Clinical Test Innovation at Semaphore, a digital health company. We collaborated with Twist Biosciences on two of our products, and today I present a talk dedicated to the implementation of Twist technology in the clinical genomics space. First, let me introduce you to Semaphore. Our company has spun off from Mount Sinai School of Medicine in New York. Semaphore incorporates a clinical laboratory connected with the Mount Sinai. We draw a lot of resources from and enjoy productive communication and collaboration with the Department of Genetics and Genomics at the Mount Sinai School of Medicine. The official spin is that Semaphore is a patient-centered health intelligence company advancing healthcare through data-driven insights. We combine high-throughput genomic testing in our laboratory with advanced analytics and use of the machine learning tools to draw the added value from the results of clinical tests. Our primary interests are in women's health, in oncology, in biopharma collaborations, and lately in COVID-19 testing and research. We combine a high throughput, high volume clinical laboratory and the latest genomics research at our company. Specifically, in the field of oncology, we offer products united under the brand of Semaphore Signal. Semaphore Signal includes the germline product, essentially a hereditary cancer test, which can be paired up with somatic testing as well. There's the comprehensive molecular profiling test looking at whole exome and whole transcriptome, as well as bioinformatics tools, of course, to analyze the results and create a relevant clinical report. We worked with Twist on two of our products, both on the germline side and on whole exome. That was a part of our cancer profiling test. A couple of slides on the hereditary cancer panel. From the total of 112 genes, we carved 17 genetic panels pertaining to specific conditions so that physicians could order the most relevant test. Our interpretation comes through both knowledgeable analysts internally and through our sophisticated analysis tools. This all is combined with a so-called Traversa platform, which allows to expand the data knowledge and provides biobanking for future testing needs. The next slide illustrates the breadth of our hereditary test offerings, all the panels, and these panels can be customized even beyond what's written here if need be. As you can imagine, behind this variety of genes and panels, there's some serious design effort on the part of clinical test innovation team. As you will know, any probe capture product goes through multiple iterations before it becomes perfect and covers all the requested targets. We have gone through two additional iterations in our efforts. First design was really almost good. Then we needed a spike in to make it nearly perfect. And finally, a few more probes, and it satisfied our needs. Similarly, we had to go through various conditions for the hybridizations and washes. And we explored hybridization temperature, the buffer composition, and the duration of hybridization. You can see the summary of our efforts on the next slide. We used both the standard hype conditions, or whatever was standard at that time, which is overnight hybridization with a non-viscous so-called original buffer. And then we used a variety of temperatures and conditions with the so-called fast, more viscous hybridization buffer. We looked at multiple parameters using Picard HS metrics, and some of that is summarized here in the table. We had to look for a perfect balance of these multiple parameters, bases mapping to the target, selected bases, right? Then targets with zero coverage. Some targets really cannot be mapped due to repetitive structure or inability to design the probes in that region. We try to minimize the percentage of duplicates naturally. This is usually the function of a library prep, but as you can see here, despite the library being the same kappa hyperplus across the board, you can see that there's some variability to capture conditions that affect the percentage of duplicates. And finally, of course, the best and the well-known fold 80 parameter that Twist is so proud of. And I have to say that these are some exceptional fold 80 values, with a caveat though, that it's just a panel of 112 genes. So it's easy to tune and manipulate and balance and redesign. So we did that, of course. But quite surprisingly, what I'm showing here is that the best condition that worked for us was a combination of the fast buffer and slow hybridization. 16 hours overnight in a fast viscous buffer did it for us, and that's what we ended up using. It may be different for your conditions and your needs. So I definitely suggest that you try what works best for you. I wanted to mention a very important point. We're a clinical genomics lab in the state of New York. You may have heard about CLIA requirements. You may have heard about CAP accreditation. Well, New York State is sort of the highest degree of lab scrutiny. Many labs choose simply not to certify their tests in New York because that is a lot of pain. It's a different level of record keeping and sample traceability and endless binders corresponding to every test that you submit. Of course, if your test is not approved in New York State, you can't offer it in New York State. And so that is a very lucrative market. Fortunately or unfortunately for us, our lab is located in New York, and therefore our first level of inspection is New York State, like it or not. So all of our tests are New York certified. On a positive note, it means a carefully reviewed, high quality test that we offer. On the negative, it takes time. Sometimes New York State Department of Health would take more than a year to issue a permission, and especially if it's not to an established and accredited New York State laboratory. This is why some of our products that are mentioned in this presentation are over a year old, because it takes that long to develop and then certify a test. So in light of that, here's an excerpt from our NGS validation, and my apologies to Twist for not talking about their latest and greatest offerings. We are looking at that, but in our certified tests, we're using the proven, somewhat old technology that was available at the time of development. Well, let me talk now about the somatic segment of our signal cancer offering. There are several options available here, depending on the client and the client's interest. We offer a wide scope, whole exome, whole transcriptome profiling. Also a narrower pan-cancer test, which is more clinically directed at the most relevant genes. And to all of that, we can add MSI profiling as needed. We spend a lot of effort perfecting the exome for our whole exome test. Indeed, since we're dealing with FFPE samples, we need high degree of sensitivity. If in the germline mutation scenario, we're looking for 50-50 allele distribution, here we need to go as low as, say, 5%. Consequently, in order to detect 5% minor allele fraction, we have to sequence pretty deep. So we targeted 250x depth of coverage. The more balanced the capture is, the less sequencing data would be needed to achieve such high coverage across all the target regions. So this slide is dedicated to the graphical representation of usable depth as a function of sequencing depth. You can see that twist exome here did better than their competitor. Again, two caveats. One is that it's the original twist exome as of a year ago. And two is that it's being compared to a competitor that is also as of a year ago. And both Twist and, for that matter, their competitor have newer versions of their exomes in the market. And I encourage you all to check them out and decide for yourself how each of them work. Another way to look at the depth distribution across the exome, or for that matter, any region of interest, is this density plot. It looks at the depth distribution in a somewhat normalized fashion, uses a smoothing function to make it look prettier. And the main point here is really in the right column, where you can notice that the depth distribution of twist is closer to normalized x equals 1 depth or coverage. That means, oh yeah, and in addition to it, if you can look at it, the peak for twist in a dotted line here is much more symmetrical around that maximum as compared to the competitor, who has a wider distributed peak with more of a tail. If you run Picard HS metrics, this comparison basically is similar to the mean and the median depth. The closer your mean depth is to median, the tighter is the depth of that distribution. And yet another parameter by which to recognize a better, tighter distribution is the famous fold 80. This all goes to say that in terms of depth, twist is rather uniform and thus looked much better to us in comparison to their competitor. Now, the important point, and really the highlight of my talk, is that both products, both the germline and hereditary cancer panel and the somatic whole exome, whole transcriptome product, were robust and rigorous enough that they received an approval from the New York State Department of Health as part of our clinical tests. This is really the highest level of approval for a lab-developed test. There are a lot of places which use twist products in an academic setting, but here, on behalf of New York State Department of Health, I suppose, I would like to vouch for twist being good enough, stable enough for the use in a clinical laboratory. As you can see, there was some difference in how long each of the products took before it received an approval. And in this case, it's roughly proportional to the product complexity. So we now proudly offer both tests in the clinical setting of our genetics laboratory. Semaphore is a digital health company, and therefore I absolutely must plug our bioinformatics, and specifically the aspect that accompanies the signal cancer testing. We rely on our core system called Centrellis for things that go far beyond just the results interpretation of a given test. This includes a very large centralized knowledge base covering thousands of variants, includes state-of-the-art genomic profiling of individual patient results, scientific interpretation of variants and discovered results, matching the patients to available clinical trials, and also, of course, the integration with large hospital systems. With that, I'm coming to the conclusions slide. So Semaphore used twist design and probe synthesis in two of our cancer genetics products, both germline and whole exome. All of that was performed in our New York State CLIA certified laboratory. After doing a few adjustments on hybridization temperature and buffer composition, we arrived to a very satisfactory targeted hereditary cancer panel. We ran the exome from Twist, which compared quite favorably to the competitor's product in the context of whole exome somatic test. And last but not least, both the hereditary and somatic cancer tests received certification from New York State Department of Health and are now offered on Semaphore menu. This work would not be possible without the contribution and help from the team, and I'm very grateful to my group and to my esteemed colleagues at Semaphore for discussion, guidance, and supervision. I close with Semaphore closing slide that has my email address on it. Look forward to future questions from you guys, and they can be either submitted online, again, my email is below, or during the Q&A session on October 29th. With that, thank you all very much. Have a nice day. Goodbye.